This is the 11th video of the PreventSARS3 video series. It's a project aims to develop nanomaterials against SARS2 pandemics and the upcoming SARS3 pandemics. I will illustrate the SARS2 adaptation and infection pattern on figures in this video. First, I would like to remind you the simple model of how other coronaviruses, normal coronaviruses in natural conditions, previous 6 coronaviruses adapted to human. So they were bat viruses or intermediate host viruses, or this could be in the intermediate host as well. That's not very significantly difficult. So they're out of the bat right now, and they're pulled into the cell by the receptors because they had very weak entrance capacity to the inside the cell. In a pocket they were digested, and few of them managed to escape their RNA with heavy damage, but some sort of repairable damage. So they found the ribosome of the host cell and formed the new adapted coronavirus, different to this one. That's the point. It should be different. So the one important difference of SARS coronavirus to compare to other coronavirus, because I say it doesn't have a sunken sialic acid binding domain, it means this. The SARS coronavirus, maybe also even compared to most of the viruses using sialic acid, capable to interact with those sialic acid on the lung surface very effectively, very tight, and very quickly. The viruses are not alive. They don't have a mechanism to move. They don't have motion mechanism like the bacteria. But with this temporary weak bonds from one sialic acid to another, it is moving. And that action motion called as viral surfing. So this is first what happens. The difference has some consequences. It has functional results of this SARS2. So by surfing on the surface very effectively, very rapidly, SARS2 is probably not being detected like other coronaviruses by the surface receptor. So it's going on the surface very rapidly, very fast, very quickly, very effectively, and then finding its receptor, that's anti-receptor, ACE2 receptor. It's a abundant plant protein on the lung surfaces, and also on the lung surface there is a lot of furin. It's a protease cutting the spike protein into two, or cleaving protein into two, so it enters to the cell. So the pandemic capacity is the result of this unique combination of superiority. So its receptor binding that perfect, domain that perfect, I'm gonna explain in the other videos more. It's not changing. There is not a positive selection site. So furin is abundant on the lungs. It's a protein available on the lungs. And this is the actual capacited reason of how the virus is causing pandemics. So the combination of all those traits enable SARS2 to become a pandemic virus. And none of the coronaviruses has these traits. So how natural this virus is, because SARS coronavirus in 2002 had no sialic acid binding effect. Its receptor binding was not that effective. It had no furin cleavage capacity, so it vanished. So how natural this virus is, and how this is possible, we haven't seen any other coronavirus with these traits if this is not engineered. So thanks for watching, and I will keep explaining in the other videos. So thanks.